George Palade, chercheur d'origine roumaine, on en a déjà beaucoup entendu parler depuis le début. Il a 34 ans lorsqu'il assiste à une conférence du professeur Claude. Il saisit alors immédiatement l'intérêt de ses recherches et lui propose de travailler avec lui. C'est une part active qu'il va prendre dans l'érudition des découvertes initiées au Rockefeller Institute. Et dans les années 50, il va découvrir puis préciser la structure des ribosomes qui effectuent la synthèse des protéines dans la cellule. Madame la Présidente, c'est le professeur Stefan de Constantinescu qui va nous retracer le parcours exceptionnel de ce scientifique.
Effectivement, c'est notre ancien président qui va rendre un hommage tout particulier au professeur George Palade, puisque son grand-père, si les informations sont bonnes, il est très très bien connu.
Merci beaucoup, Madame la Présidente, Monsieur le Secrétaire perpétuel, chers membres du bureau, chers collègues, Mesdames et Messieurs, je vais me permettre de présenter George Palade. So, George Palade was born in Romania in 1912, actually on the 19th of November. He was born in a city called Iași in Romanian, or Iași in French. It was the capital of the province of Moldova, which in various times of the history of Romania included the region that is at the border with Ukraine, which is now called the Republic of Moldova, which is an independent country. But Iași remains part of the Romanian Moldova. His father was a professor of philosophy, and his mother was a teacher, a primary school teacher. So since his early age, he was deeply involved in reading, in discussing with his parents, and received a very humanist culture. Even in his primary school, all his teachers were amazed by his intellect. He was really an exceptional individual, and he went to a very good high school south of Iași, in a city called Buzău in Romania, where there was one of the best colleges in Romania at the time, and obtained his baccalaureate, which was at the end of high school, with the maximal score, which was almost impossible. At the time in Romania, as far as I read, the high school and the baccalaureate were more exigent than in the best high schools in Paris, for example, Henri IV or Ville-le-Grand. This is not the case, unfortunately, today. But at the time, somebody with a maximum score was unique. So he was received at the medical school in Bucharest at the Faculty of Medicine in 1930, which belonged at the time to the University of Bucharest. At this time, it's a university itself of medicine and pharmacy that carries the name of Carol Davila, a French military physician who founded the medical training in Romania. In the medical school, he continued his proof of excellence, being the absolute top of his cohort, and since he was a student, he began doing research in the medical school in Bucharest with 3 professors in the same time, and that was very unusual. He had a talent for drawing and for space orientation, and could memorize all the peaks and rivers of mountains in Romania without any effort. So he worked with Francis Ieriner, who was a professor of anatomy and specialist also in evolution, who created the Institute of Anthropology in Bucharest, with Grigore T. Popa, who actually was returning from Rockefeller at the time, after having discovered the Porter circulation between the pituitary and the hypothalamus region. So this was a very high-level scientist that worked for many years at Rockefeller and well-known in endocrinology, and with a French professor of biochemistry, André Boivin. At the time, the medical school in Bucharest, when they didn't have enough competence in an area, they were recruiting abroad. So they recruited André Boivin, who was a very high-level scientist and stayed in Romania until 1933, between 1928 and 1933, so that was really a chance for George Palade. André Boivin went ahead afterwards and worked at the Pasteur Institute and at the University of Strasbourg and discovered the glycolipid nature of blood surface antigens, so it's a very high-level scientist. So this student, George Palade, was on one hand a very high-level anatomist, becoming one of the key students achieving in anatomy, but on the other hand, he was studying biochemistry. So he coupled these two fields, which are not usually the passion of the same individual. So either you are given gifts for anatomy or for biochemistry. For his MD thesis, which he obtained in 1940, so this is the thesis equivalent to the end of the medical studies, he never had a PhD. That's it. That's the maximum diploma, MD, in 1940. He decided to work on the kidney of the dolphin as a subject, which was found a bit exotic at the time, because there were a lot of dolphins in the Black Sea, and because the kidney of the dolphin is composed of multiple granules, each functioning as a kidney in itself. And he did a study of microscopy and serial sections, demonstrating that the urinary tube and the Henle loop have a length and a structure which reminds of animals that live on Earth and not on sea, and proposed that this went to the sea after being first a mammal of Earth, of dry weather. And of course, everybody was a bit worried that he spent a lot of time on the dolphin, but eventually he finished his study. This is still today in the Department of Anatomy. This is the medical school that I also studied medicine in, and we all have been amazed by this model. And this is his dedication to his professors at the end of his medical studies. So between 1940 and 1945, he became a faculty member in the Department of Anatomy, arriving to associate professor, and then he also did internal medicine's residency and became an internal medicine specialist at an old hospital in Bucharest, Colentina Hospital. As you know, the Second World War resulted in the fact that the Soviet army occupied Romania, and there's a couple of 100,000 soldiers there. Research was not the main priority, so he decided to go and study abroad and went to the United States. He was lucky enough to escape before the borders were closed. And first he did a visiting fellowship with Professor Robert Chambers at the New York University. But during the couple of months he was in New York University, he had the fortune to attend, by chance, a seminar from Albert Claude, who was invited to the NYU to give a seminar on his studies. And when he heard Albert Claude talking about fractionation, ultracentrifugation, and coupling it with electron microscopy, he immediately went to Albert Claude and asked to come and work for him. And in a few months this was done, and he moved to Rockefeller. And at Rockefeller he was received by a group of exceptional individuals, Dr. James Murphy, who was actually the head of the Department of Experimental Medicine, Professor Peyton Rouse, the discoverer of the Rouse sarcoma virus, and Herbert Spencer Gasser, who was at the time head of the Rockefeller and just got the Nobel Prize in 1944 for neurosciences of physiology. Now, since Palade was a great anatomist and knew very well the work of Johann Laurentius Gasser, who discovered the trigeminal ganglion, and apparently Herbert Spencer Gasser was distantly related to the Gasser of anatomy, everybody really received him with a lot of acclaim, and he started working in the lab of Albert Claude, together with Keith Porter and later on with Philip Sikevich. At the time, the biggest problem they had was that when the cells were submitted to ultracentrifugation, the force was so high that what they would detect by electron microscopy of the fractions was a lot of cellular debris and pieces of organelles. When they looked at cells in culture, the image was different. So nobody succeeded to observe the same structures after ultracentrifugation versus after looking at cells in culture. Tissues were not possible to be studied because they were too thick and there was a problem of penetrance. And so George Palade remembered that when his mother and his aunt were making jam and marmalade and wanted to keep the fruit intact, they were adding a lot of sugar. So he added 0.88 molar sucrose to Albert Claude's buffer, and the miracle happened. The structures that could be observed were of exceptional resolution. And this is the first paper of Palade describing the mitochondria, describing the outer membrane, the inner membrane, the intermembrane space, which we know how important it is, and he correctly predicted that the cristae of the mitochondria come from the inner membrane, and it's a way to amplify the surface of the inner membrane. He also observed the proteins that were there and predicted they are the enzymes and the transporters that help mitochondria accomplish the function of phosphooxidation. And then the next problem was how to fix samples for electron microscopy. And because he was really a good chemist, he tried many things, and in the end, he came up with this solution to use a tetraoxide of osmium, but buffered with veronal at pH 7.3, 7.5, and that all of a sudden allowed to have very good quality of EM. And so he discovered these granules, which were called initially Palade's granules, and then he called them ribosomes, and saw that they are present both freely in the cytosol, but also on the membrane of endoplasmic reticulum, who had been discovered by his colleague Keith Porter. They discovered together the transitional elements of ER, the smooth ER, where steroid hormones are produced. And with his postdoc, David Sabatini, a future colleague that then went to NYU, they actually showed that the microsomes of Albert Claude contained endoplasmic reticulum plus ribosomes attached. And then with Sabatini, they actually showed that the protein that is made by the ribosome gets into the ER. And they also showed that the ribosomes are the same between the free ribosomes and the attached ribosomes. What is different is the protein that happens to be translated. And that led then to the whole signal sequence and the whole field of targeting of proteins in the cell. Now, the many electron microscopy pictures that Palade took showed a lot of vesicles in all types of cells. And with Herbert Gasser and Sanford Palade, they discovered what is today the structural basis of synaptic transmission. So they had the surprise that in the synapse, the presynaptic nervous terminal contains lots of vesicles. And once the influx came, those were discharged. So they actually showed that the neurotransmitters were in vesicles that were already charged, and then when you transmit the signal, what happens is that all these vesicles fuse with the membrane, deliver the mediator into the synapse. Interestingly, Palade never made a big deal of that and always said that this is a side track, and he was not very proud of this work, saying it's a narrow field of neuroscience and wanted to only work on other things that are more general. Now imagine today, if somebody discovers that, it's the whole subject of the lab. So he then discovered the principle of vesicle traffic in the cell, so that the macromolecules are not freely diffusing between an organelle and another, but the whole thing is going through vesicles that are coming from one organelle and go to another and fuse, and that is true even in exocrine, endocrine organs. Here is insulin vesicle that is before and after delivery, and of course in the synapse. And so he was, until the beginning of the 50s and 60s, one of the leaders in the electron microscopy. Albert Claude had left Rockefeller in '49, so he was head of the lab after the departure of Albert Claude, and they really described the endoplasmic reticulum, the rough endoplasmic reticulum, all the Golgi compartments, the trans-Golgi network. And he had shown in 1960 that ribosomes synthesize proteins. And then he was fascinated by this subject, and that is, what happens with these proteins? Where do they go? And how do proteins get out of the cell? What we call today the secretory pathway. So he chose, and that is the secret of success, he chose the right tissue. He chose the exocrine pancreas to study protein secretion, because obviously it's a specialized tissue that makes a lot of protein. And initially, with one of his collaborators, Lucian Caro, he discovered that the enzymes of the exocrine pancreas are stored in vesicles, in condensing vesicles that come from the Golgi. And he proposed that the Golgi is really a hub for protein dispersion in the cell. Before, Golgi was thought to be perhaps an artifact or some kind of derivation of the endoplasmic reticulum. And then with James Jemison, George Palade did the experiment that brought him the Nobel Prize. And that is, at the time, people were trying to purify everything that is in the ER, purify what is in different Golgi segments and vesicles, and it was very difficult to make sense of the enzymes because it was easy to contaminate one fraction with another. So George Palade came up with a different idea. He basically took slices of exocrine pancreas and then labeled protein synthesis with radioactive amino acids, pioneering the technique which we call today the pulse chase technique. That is, you incubate the slice of tissue or cells in a medium without one amino acid, for example leucine, then the cells are starved, you add tritiated leucine, all proteins incorporate leucine, everybody in the first minutes will be labeled, and then you add excess non-labeled leucine, 100-fold more, so that that labeling stops, because the non-labeled will take the place of the label. And then he did autoradiography and electron microscopy on different time course points. And what they show is that initially, after a few minutes of labeling, you see the labeling, this black dots on ribosome and endoplasmic reticulum, then inside the endoplasmic reticulum, then in the Golgi, then in immature vesicles, then in mature vesicles, and then outside the cell. And it is this way that they discovered that the cell is polarized and accomplishes a vectorial transport from the ribosome to the ER, to the Golgi, and different stations of the Golgi, and then to the extracellular part. Now, this pathway was then exploited by continuators of his work. For example, Harvey Lodich discovered that it's the same pathway that puts membrane proteins at the surface, or where they will not be freely floating in the plasma, but they will in the soup, but they will be anchored at the surface. So in 1973, after all this work on electron microscopy fractionation and then his experiments with secretion, which pioneered the whole field of exocytosis, which is the exact inverse and endocytosis of Christian de Duve, he went to Yale in 1973 because he thought that time has come for these fundamental discoveries to be exploited by medicine. So he wanted to interact with pathologists and clinicians and cancer and other clinicians. And he created a section of cell biology that then became the Department of Cell Biology. And in that stage, he was working on membrane biogenesis, membrane trafficking, capillary biology, and link to medicine. But 1 year after, he got the Nobel Prize in 1974 with Albert Claude and with Christian de Duve, and that gave an impetus to invest in other areas, and one of his main areas then was the cell biology of endothelial cells and capillaries. Because he was a great anatomist, he knew how capillaries work in all tissues, and early on in the 60s he had discovered with Weibel the Weibel-Palade granules, which are structures, tubulovesicular structures that host the von Willebrand factor, which is a very important protein in coagulation, and it's at the center of hematology. And then he went ahead and with Marlene Farquhar, who was a specialist of capillaries in the kidney, they discovered what we call today the tight and adherens junctions, which in itself is a chapter of cell biology. And then the ultrastructure of the renal glomerulus. They used ferritin as tracer in capillary studies, and described how disease in the kidney affects the ultrastructure of these organs. And with his two Romanian co-workers, Nicolae Simionescu and Maya Simionescu, they really discovered transcytosis. They presented evidence for transport of small molecules in continuous endothelium via plasma-lemmal vesicles, which are vesicles he discovered before, and really changed the way people think about transport in the capillaries. Now, he was also a creator of many, many institutions all over the world, but I'm going to just stop at one, which is an Institute of Cellular Biology and Pathology, created in 1979 in Bucharest, under the leadership of his two co-workers, Nicolae and Maya Simionescu. And at the inauguration, Christian de Duve, Gunther Global, and several others were present, and it is where Christian de Duve gave that speech that is now in the book, in the album, about the thrills of science. Now, at Yale, he developed this department, and really all his life, George Palade encouraged many, many young scientists, so he was one of the most stimulating people to really help careers and science of younger people. He recruited many, many people in that department. For example, Ari Helenius, Ira Melman, Petro de Camilli. These are people that are very, very important in the modern cell biology. And in 1999, his direct successor, Gunther Global, that took over his lab when he left in 1973, received the Nobel Prize for Medicine alone for the principle of addressing proteins in the cell to different addresses via signal sequences. And it is this work that started with Palade and Sabatini and continued with Global that allow us to know today exactly how a ribosome that synthesizes a protein that is destined for being secreted or being on a plasma membrane is going to be attached to the reticulum, how it's translocated, and then the whole chapters of folding, glycosylation, and the whole world of the secretory pathway. Another one of his Yale recruits and colleagues, James Rothman, together with Randy Schneckman and Thomas Sudhoff, got the Nobel Prize in 2013 for the genetics, the exact proteins that address the vesicles in the cell, and that culminated really the legacy of George Palade. At age 78, he decided that he didn't have enough challenges and took another job, which was to be Professor and Dean of Science at the University of California, San Diego School of Medicine, where he was fully active until 2001. He became Dean for Scientific Affairs, created the Department of Molecular and Cellular Medicine with Marlene Farquhar, recruited over 30 faculty in cell biology, such as Don Cleveland, Peter Novick, and many others, who are really prime stars of biology today. Attracted the Ludwig Institute for Cancer Research San Diego branch with the help of Christian de Duve, who had done it earlier in the 70s in Brussels, and also created the Howard Hughes Medical Institute station at the University of California, San Diego. So for his entire life, George Palade stimulated and helped young people all over the world. I have to say that I have had the honor to know him personally, because when he left Romania in 1945, he came to see my grandfather, who was a professor of virology in Romania, formerly at the Pasteur, and he gave him a letter of reference, and my mother, who was a child, remembers that her father told her, "This is the brightest person that this faculty ever had, and you will hear about this person in the future." And clearly, this was a very, very great achievement. And so in University of California, San Diego, the names, he really, the building that he created is now called the George Palade Laboratories for Cellular and Molecular Medicine. And at one of his last appearances, Marlene Farquhar cites that he said that for him, the names of all the collaborators that he had in his scientific life, when he reads them, they read for him like a poem. So George Palade really helped a lot of scientists all over the world. So I'd like to end by acknowledging several people that were very important for the book, for the part on George Palade. Of course, Randy Schneckman, who wrote a piece of it, Maya Simionescu, Pierre Courtois, Emil van Schaffingen, Daniel Godin, Radu Iftimovic from Romania, and George Casimir, who had the idea of creating this book. And I want to just take 1 second to say that at the end of my medical school, I worked with a historian of medicine, Radu Iftimovic, who wrote a book about Palade and went to the United States and took an interview. And I had the privilege to hear all the audio cassettes. And so I know exactly what he himself said and what's now in the book. So that's why I accepted to give this tribute to George Palade, because I have been exposed to this discovery very early. Thank you.